Hey, do you know where?
Hi, Berg. Hello, everyone. Yeah, good. So,
I'll give it a couple more minutes, then I'll kick it off afterwards, just to
sure.
let people trickle in. Yeah, let's give it another minute. We have like 120 registrants, so there might be people that join in a couple minutes.
Yeah, I think I can use the time to test the screen sharing.
Go for it.
Yeah,
Yep, can see,
nice. Yeah.
can see.
How about now? Can you see the whole,
We see
screen?
the whole page. Yeah,
That's nice. Yeah.
yeah. Okay, let me kick things off slowly then. I'll quickly share my screen as well. I'll just take a couple of minutes. Hello, everybody. My name is Berkey Bukudek. I'm the CEO and co-founder of Superbio. And so I'll show you. So Superbio is essentially a low-code platform and marketplace for biomedical AI. Our goal is to really accelerate scientific research by making AI models accessible um, and usable by the wider scientific community. Um, if you can go to app.superbio.ai, it works like really any other app store. You have a bunch of apps that has been sorted by recently added, most runs, most stars. You can basically look for anything you'd like. So let's say the most popular one, usually AlphaFold. You can click on one of these apps. All of the apps have descriptions provided by the researchers themselves, and you can put in some parameters, put in some input data, submit it on our CPU or GPU, and once your job complete, you get an email. You can see the result within the platform. Hatian will do a better demo than I will. And Hatian is the main author, the lead author of one of the most popular apps on our platform, which is scGPT, which has three different paths. And it's why we had reminded him to give a talk on how to build a foundation model in biology, what are the future, and what does scGPT do, and what's the future for scGPT. So without further ado, Hati, thank you for being here. Why don't you take it away?
Thank you, Berg. Thank you for this welcoming introduction. Let me try to share the screen. Uh, yeah, it's working, right?
Yes, can see.
Yeah, great. So yeah, really honored to be here. And Superbio has really been a huge helping hand for us, try to expand the accessibility for our model. So in today's talk, I'd like to present this work as GPT created by us. And we're a group of researchers in University of Toronto. And I will start with the introduction to the method itself, and I'll try to also see some application demo around one particular thing we call zero-shot reference mapping. And hopefully this can give you some idea about how to easily use a model and the model itself. Yeah. Yeah, feel free to interrupt anytime in between and ask questions. Unless you've been living in a cave for the past years, other than that, you probably know that the ChatGPT, Alpaca, or the Gemini thing is promising generative AI age that in the past year, we really see this huge surge of large language models that they are pre-trained on huge data set with very high diversity and achieved remarkable success in various domains. For example, we all know that GPT-4 has really surpassed human a lot of those benchmarks and like very difficult tasks and exams. Especially what we see is that we see the combination of this large scale diverse data set and the way of pre-training transformer. And so when you merge them together, this becomes this very promising approach for developing a foundation model that can learn the knowledge of certain domain and then apply to various tasks and applications. So how about that for biology, specifically for single cell biology? So for people who are not familiar with this idea, like single cell is like, is a kind of like sequencing technology that not only sequence, the genome of a human body, but sequence like on individual cells. So this really give you high throughput and diverse input data. And this analogy, while texts are made of words, cells can be characterized by genes. So this analogy inspires us to explore this foundation model for single cell. In the long run, we kind of hope that you have all the diversity in millions of sequence cells, and if we can learn from them how the gene expression, differential expression in all the cells, maybe we can build up a model that works like a simulator of molecular biology, right? And you can use to potentially understand different questions between normal and disease conditions and things like that. Particularly, we think is feasible based on two considerations. The first is that transformer has been used in language, but things we like analogously like this one, we also know that a transformer can be suitable to learn interaction between genes and other molecular tokens. You can use a self-attention mechanism to model like the regulation between genes, right? And the other thing is about the vast scale of sequencing data actually is growing exponentially. And on the right hand here, I show you said this is a screenshot from CellXGen, which is a platform supported by the Chan Zuckerberg initiative, and it's like a portal. They collected different cells of humans and other species. Particularly, this is from May, I think. So it already had like 33 million unique cells and across different tissues. And nowadays, I think it's on the order of 50 million. So you can see the growing of the data in this community. So based on this observation, and so we try to build the scGPT model, which is supposed to be we wanted it to be a single cell foundation model we pre-trained it on 33 million normal human cells, actually from the CellXGen data. So this is like the pre-training stage. You have all the diverse cell types from across studies and train them in a large model. And then you try to fine-tune the model for different tasks with task-specific supervisions. So this is just some high-level diagram I show here. I want to give you a idea of how the thing works. And so in the pre-training, we kind of look at the data. In the 33 million cells we used for the pre-training, it covers actually 51 different organs, and actually I think more than 400 studies. And at least you see the diversity. There are some things that you can observe is that the tissues, the number of cells in different organs and tissues, they don't come in very balanced way. So you have a lot of maybe one third of the cells are like brain tissue, and then you have various of cells like covering other major organs. So that is a hint that we know that we kind of have the diversity trained model, but the performance may not work balancedly for all applications. You can imagine that it might perform a little bit better in brain related questions or in blood or immune cell related applications. So a high level view for the fine tuning, we created like different self-supervised and supervised objectives specifically for the fine tuning stage, which is try to adapt the model for different scenarios that we always see in single cell applications. For example, like biologists might want to clustering the cells, so you need very good embedding of the cells, and you want to batch correction. This is something specifically for computational biology, for this sequencing data. It's like, you have all the cells that are you supposedly there are two cells that are from the same cell type. They are all B cells, but one is sequencing in one center in one machine, the other is from another center in another machine. So there will be some technical difference between the two cells. So we wanted to remove those technical difference. We call this kind of task like batch correction. Like batch here meaning the technical batch of the sequencing machine and other things. And we want to do integration of all these different studies, and we want to find the network gene regulatory interactions between genes. And particularly, we kind of want the model, actually, we prove the model is performing very well for tasks like perturbation prediction, which is like given a cell, and supposedly you do some maybe CRISPR-based knockout of some gene, and then how the cell will respond to this stimuli. So so those are various different tasks. You can actually find our tutorial for this task in our paper, in tutorials in the GitHub, and you can find the description for how we do this in our manuscript. So today, actually, the thing I want to highlight is about some new things we are currently exploring, and we have recently put out resources and tutorials on GitHub, on our GitHub repo. This is about the zero-shot application of using scGPT. So I just want to briefly have one or two sentences to mention the rationale of this. So the model itself is relatively a large model. We try to keep it smaller than the other large language models you see for natural language, definitely, but it's still something like 53 million parameters. We're actually also trying to build larger ones. But you see, it can be a challenge for a lot of researchers, biological researchers. They are not very familiar with GPU, or they don't have access to a large amount of GPU computing resources or time. So the thing about zero-shot application is that sometimes you don't need to do fine-tuning. Then you don't need to need huge amount of GPU. You can use a model off the shelf. You can use a pre-trained model to compute some embeddings of the cells or embedding of the genes, and to give you some insight of applications. So they can be much faster and much more accessible for a lot of researchers around the world, like biologists who don't usually work with GPUs, right? And the UMAP of zero-shot embedding, here we just show some examples of how we apply the zero-shot application of scGPT, particularly on two disease data sets. So I think the first one here is a COVID-19 data set, and the second one is about a lung cancer data set. So we see that the way we do this is that the first row here is like we use a model, the pre-trained model, and feed the model with this new cells from this data set. And then the model will generate the embeddings based on the expression profile of the cells. And the embedding is particularly like a continuous vector of 500 something dimensions. And then we use a method called UMAP to project the embedding onto low dimension. The ideal case here you want to see that if the embedding works really well, is the embedding captures the different biology in the cell, and also mitigate the technical difference, of the sequencing machines, things like that. And then you expect to see that for this column, for the cell type column, you exactly they are clustered very well by these colors. Since each color is annotated by the authors in this data set, and they say these are where's the color? Yeah, this is the color for the cell type. You see the blue one is for this AT2 cell, which is some short name, and that the green one is like macrophages, and then that's one hand. And the other hand, we want to see a mixed color, like you don't want the model to tell so much about the technical difference about the batch, because something is sampled from 10x, something is some cells are from one sample, some cells might from another one, and using different technologies. The thing we observe here is that actually the zero-shot mode can give you a pretty good result, like shown up here, right? You can see the different cell types that are colored and clustered very well for the major cell types. And admittedly, this is a very challenging data set, and you can see, there are definitely some space to improvement for those cell types. This is actually, I did show the other thing. This is actually a pretty good score, at least for this data set. I also tried in more conventional ways like Harmony or scBI or Seurat. I think scBI performs the best among those conventional methods, and its around have a score we call it average bio scores. Basically, the higher the score for those two, the better you have. So for the conventional method like scBI, it's around 0.5 something. So it's very close. You can see this, zero-shot mode generate pretty good embedding. And similarly for the other task. The second row is to, in a fair comparison, people would also try PCA, which used minimal training, but ideally people don't consider it to be a trained method with heavy compute. So the zero shot and PCA actually they can complete in similar times if you have a consumer and consumer level GPU to help you to do this zero shot. So in this similar time scope, you can see that it most of the time actually performs at least better than PCA. So it shows that the zero shot embedding can actually be a good tool for researchers. Yeah. So that's the thing about we call it the integration. The next thing is that once you have the embedding, the researchers would like to use the embedding to. Sorry, is there a question? Oh yeah. The question is about how do I deal with the fact that a single cell data is tabular data and does not have semantic relation between the expression values compared to synthesis. I omit the details, so I think the question is about whether the sequence makes sense in the. Okay, I didn't have the slide in the pre-training. So the tabular data tells you that the single cell is more like for gene A, you have this expression, for gene B, you have the expression. So in a sense, this input is less like a sequence, it's more like a set. Actually, transformer can deal with that. The self-attention in transformer, they don't have the, so it's outer agnostic. You switch the order of the input, the transformer will generate do the same thing. The thing people use for language model, actually, they inject a position embeddings, which is another additional technique to make the transformer work with sequential data. So actually for us, if we only talk about the difference of order, the difference between a set and the sequence, actually it's not a big deal for transformer model, and we can use that in the work. Yeah. Another map in the pre-training. Another question about does scGPT only work on single cell RNA-seq data, or it can be used on single cell analysis from multiplex imaging data? Oh yeah, that's a great question. Actually, the first part, we do not only work on single cell RNA-seq. The pre-training is only RNA-seq data, which is like actually single cell RNA and single nucleus RNA in the pre-training, which is more or less like they are the data type with a great abundance so far. And we tried some applications on multi-omics integration, which including ATAC-seq and site-seq, things like that. The challenge for the model is that we just need to make some adaptation to inject some new tokens to the model that representing the ATAC or site-seq data during the fine-tuning stage. So you can find that in our tutorials and in our GitHub. But the second part of your question, which is really a great question about multiplex imaging data, we currently don't support that. So imaging can be a really different modality than all this sequencing. I'm imagining you're talking about something like spatial omics stuff, right? You have a paired image and some sequencing data, which is really interesting. We currently don't support, but actually it's in our line, and we wanted to release some model doing that in the future. Yeah. Yeah, let me know if I answer the question. So if I may continue, yeah, I think the time's still okay. The thing I want to mention that here, in the previous slide, we saw the cell embeddings in the zero-shot mode. And then, researchers can use that to label your cells, right? To do this cell type labeling in the reference mapping manner. So for example, here, I think this is the same data set of the lung cancer data set. And we try to split the data set into reference and training based on they're from different samples. So we put some samples in the query set. So the idea is that we try to mimic the setting that, the people might have a reference data set where they already label the cells with some annotations. And then we have the cell embeddings. So we can compute who are the nearest neighbors of all your query cells, right? And then we propagate the labels from the reference set to the query. And this can work like a very efficient way to classify, new cells to different cell types. And the whole thing is called reference mapping. This workflow is called reference mapping in the domain. And we see that it works pretty well. Actually, the zero-shot setting generate very good reference mapping results, in this evaluation metrics. Then some other existing techniques that are specially designed for reference mapping. And also it's very close in most of the cases, at least for this 2 data set. We didn't really, test a lot of data set, but at least for this tool, is performing very close to fine-tuned model. Yeah, so I think those are the theoretical part. And okay, there's one slide more about theoretical part, which is about the same setting, but you can imagine you can also use a reference set that contain millions of cells, maybe on cell exchange, like the 33 million cells. And then we can use a model to compute the nearest neighbor between your query cell and all the cells in a huge atlas, right? You can have this model to generate embeddings for all of them, and then you store the embedding for the atlas, and then you can compute the nearest neighbors for any query cells. And we actually also recently, provide an example doing this in our tutorial in this link. Thanks to the engineering from the Meta, they provided this very nice toolbox called the Face toolbox. We can really compute the nearest neighbor really fast and make this whole process very computational efficiency. And so, for example, imagine you have some 4,000 query cells. If you have a GPU, you can compute the query from this 4,000 to millions of the cells in the cell exchange in just 100 milliseconds. And then you can get the labels, and you can propagate the labels and to label the cells. And that's like milliseconds on GPU, and also very fast on CPU, actually 7 seconds for 4,000 cells. Yeah. This is a competition step for the nearest neighbor. Yeah. So that's about the theoretical part. And let's go to some demos. First thing is about Google Colab, and then we can show the great thing Cell Exchange have offered about the web tool. So yeah, if you go to I just wanted to point you away where to find this Google Colab. If you go to our GitHub link, and here you go to the folder of the tutorial, you can see all the other tutorials about different applications I just briefly mentioned. There's a quick question from the chat. Are these models pre-trained mouse data? No. So far, these are only for human models. We do provide different models. One is called for all human cells, and some other organ-specific models for different organs, but not yet for mouse. But we actually see a lot of this request, and we are also wish to plan to release one like that soon. I think the technical part is actually, we think that will be similar. Yeah. Oh, for batch normalization, this is a great question. I think I forgot to mention this. Okay, I need some additional time for it. I think it's really worth mentioning this. Yeah. So there's a question about this reference mapping, about the batch correction. Yeah. So that's the thing. All the things come right from off-the-shelf pre-trained model. The thing we observe is that the model itself, the generated cell embedding, has some effects of doing batch interaction. You can see it ready to get some batches, but I would be totally honest here. It's not doing perfectly. There definitely are other methods specifically designed for batch. Actually, there's also our fine tuning is that there are also a way in our fine tuning specifically designed for batch effect removal that can perform better. But the thing here is that it's really interesting the pre-trained model can do something. And the one side is that I can explain that why the model does this. It's because you see all the different cells automatically from different studies, and we are trained them in a self-supervised way. So the model we also have some techniques in pre-processing to mitigate the batch effects a little bit. But in general, the model need to understand, okay, those are the same cells, and to learn a very good biological representation for them. But the other side of the story that it's not doing perfect, right? You can still see some batches, and that's in a way that's expected, because you can also think about it in that way that the pre-training process, we do not design them especially for technical batch effects. And the pre-training is, in general, a self-supervised learning for the model to reconstruct the gene expression in the cells. And so you can think about it in the other way. Some of the batch effects are actually very important signal for the pre-training stage. For example, the samples come from 10x they're always different. The distribution of the gene capturing rate in 10x will just be different from other techniques, for example, like SmartSeq, right? So this kind of signals, this kind of batch effects can be important signal in the pre-training. So the model itself are not guaranteed, are not expected to mitigate those batch effects. So to answer the question, the batch normalization is not for this. If we are talking about this zero-shot application, the batch effectation, we don't apply other specific techniques on expression level or cell level. It's just something automatically came from the model. But the other side of the story that sometimes you need to be cautious about applying the model on samples that are from different sequencing machines. Yeah. Okay, I think there are more questions. I'll try to answer them later, but given the time, I'll start the demo first. Yeah. About the demo. So like I talked about, you can find the tutorials for a lot of applications, and specifically, we have a demo here in the zero shot. There's a new demo, which is about zero shot reference mapping I just talked about. And if you click here, you can either download, install the package yourself and try to run it, and also you can open that in Colab. If you open that in Colab, you see that here. I think I just run the model, but I think I could try to run out again. And before you run, if you open this yourself, just remember to change this to T4 GPU. So the next thing is that we are quite excited about this because we recently upload the whole process of installation. So right now you can really easily install the model itself, and so you can see that you can naively work in Colab, and you can see the all the process. And I will just briefly go through the cells because I think that can give you the idea how the thing I talk about, how they actually runs, right? So this is just about import the thing, and then we provided the sample data. This is the COVID data I just showed you, and then you have the training data, you also have the test data. We call it train. This is like the reference set. So the idea is that the model can generate the embeddings for both partition of the data set, and then it propagates the labels from the training data to the test data, and we can evaluate a little bit on how well it works on test data. The important thing is that you need to keep these two columns. The first column is an optional one. It's a column in the training data that the label of the cell types in your training, like where are they, right? And the second thing is about the gene column, which is really important in the end data, because we need to have the correct gene names. We're going to match the gene names from your new data to our training vocabulary. So you need to provide the column of the names, provide the column names for us to find the gene names in the column, right? And then there's a one-line wrapper that lets you to embed the data to generate you cell embeddings. And you can see, it runs pretty fast today somehow. It finished in 34 seconds on this T4 GPU. And then, yeah, this is for the test data. And I think I can just go through this pretty fast. And we can skip this block, I think. Yeah, this is just some code to concat the data together, but this is just for visualization, right? And yeah, so after the concatenation, we just visualize them all together. You can see that this is a UMAP computed by the embedding, and you can see the query, your query cell, and the reference cell. And you can see where are they in the cell types. So the cell types, so this is something we visualize here, but you know that the cell types for the query is unknown to the model. So we mark them as to be predicted here in the visualization. So as a human, right, if we think the cell embedding is good enough, we may guess, okay, this must be some macrophages, and this might be some, this AT2 cells. And there are some cells that are quite tricky around here because they're in, relatively speaking, low number of abundance, right? Lower abundance. And then we do this reference mapping, which is basically compute some similarity between the cells. You can also use a phase model to compute your similarity. We provide an example here. And after the computation, you find the k equals 10, you find the 10 nearest neighbors for each cell. And the most, you do this majority voting between your 10 nearest neighbors. And the majority voted cell type will be the predicted cell type. And that's how we do this. And we can see, we evaluate the accuracy, those things. We can see the accuracy is pretty high. And we can see this is really some challenging case. We can see the recall and the macro F1 is relatively lower. And this is particularly because, this number of cells, which is really challenging here, right? And you know that the precision here, the precision recall and the macro F1 are computed average of all the different cell types. So even the cell types with relatively very small number of cells, they should share they would influence the score a lot. So that's the thing. This reference-based mapping can do pretty well for large, for cell types that are common great abundance. For example, this AT2 cell we just see, right? You can see the precision is pretty high. And for macrophage, it's also pretty high, right? And obviously, it can be challenging for this very small very subtle cell type of a small number of cells. Yeah. And for this kind of scenario, you can go to fine tuning and try to see whether you can improve the performance a bit more. Yeah. Actually, it runs, yeah, now it's running to this tab, right? Yeah, this is just to show the result on the other one. I think this already give you some ideas of how the demo works. Let me see if there's any question related to demo. Yeah, I think that looks fine. So that's it. So the next thing is about the file stream. Sorry, about this wonderful web app developed by created by Superbio team and help us. We just see the Google Colab. The thing is that first, sometimes you don't even want to work on Google Colab to write your own code, right? And actually, Superbio gives this very nice app that help you to do this. I just wanted to quickly show the same data set and how you can work in this web app. Actually, this web app runs in a different setting, right? The other one is that we try to mimic, you have some reference, you have some user provided custom reference data set, and then you have the product data set, right? So this one, this is the second mode I talked about in the presentation. This is about reference mapping using cell embedding, but I think it's not mentioned here. But this is the reference data set you use is a whole cell atlas from cell embedding, from cell XGen. So it's 33 million cells. So now the workflow is that the workflow hidden in the backend is that you upload some data, you provide the this is optional. Maybe you don't want to provide the cell type column, right? Since you don't know that yet, but you can provide the gene column name. So you upload the data, you provide the gene column in your data. The model will process your data, and then it will get a cell embedding, and then we'll compute the nearest neighbor between each one of your provided data and the 33 million cells in the large cell atlas, and then try to propagate the label from the cell atlas to your data itself. Yeah, I think I can go. So you can go to this web URL and find this web tool. The only thing, so this part of these, I previously run 3 runs, and you can see the history here. And if you want to run a new run, you go here, and this describes the data structure. You just need to have a H5AD file, which is usually the AND-data file format, right? And you can upload your file here. Here I actually uploaded one. I can re-upload that thing. Yeah, this is the COVID sample. Oh, yeah, it's maybe I can refresh. Yeah, so let me try to upload the COVID sample. You can see how it works. The next thing is that you can preview the AND data. You can have a preview and know what the column you should put in. Usually it runs faster for me. Yeah. So here is like the VAR, which in the AND data is basically the kind of pandas data frame that are corresponding to your genes, right? You have all the name of the genes, and you have specifically we want to know this. We want to know this gene name column. So I will copy this. So this is a column name of your genes, right? The model later will go to this column and extract these names and try to match them to the vocabulary we had in the pre-training, right? And yeah, so the thing is that you have this preview and you see whether there are some interesting things, especially like this gene column. There's also this preview for the pandas data frame corresponding to the different cells. Okay, so that's a gene name. Wait, I didn't copy that. Oh, there's something weird with my computer. Give me 1 second. Okay. Yeah, and then you can just click submit job and run on the GPU. I think at least so far, the Superbio team is really nice, offering free GPU orders and uploading the data. I guess maybe a lot of people are actually using this. Yeah, So you can see all the data, so preparing, and then we'll go to the training. And we don't need to wait for this one. It actually finished pretty fast. I think 3 or 4 minutes, because I tried this like yesterday. We can see one of the example I just tried yesterday, I think. And this one I also uploaded the original labels because I want to see, you know, how well the thing compared, the predict label compared to the original one. To me, it actually works pretty well for this data. You know, you see the predict label. These are, you know, various labels from the cell exchange. So the name of the cell type are not necessarily be the same set as you see in your provided one, right? But you can see it matches well for most of the ones. It guess this one might be CD8 positive T cell, but at least it's a T cell. And for this one, it's really accurate. It's CD4 positive, and you know, macrophages, classical monocytes. Just a notice. This works like in my test. I also tested this on our data set. I mean, you know, this setting. This is one of the cases works really well. So far, the issue is that the number of cells in the cell exchange are not balanced. So you see a lot of cells are immune cells and are brain cells. So actually, so that explains actually this mode, this reference mapping with cell exchange will usually run pretty well for brain samples or blood samples. But you have other samples. Sometimes you test with pancreas samples or things like other things. Maybe you will not get as suboptimal. You will get some suboptimal results because it's just that the cells are not in balance. You know, relatively not in balance in the allos, and you can get the model might be confused in finding those neighbors. There are multiple ways. We're actually trying on this to deal with this. Actually, this actually related to my concluding slide. I just wanted to mention this. Those are a lot of things that we're really interested to try to even increase the quality of the model and of the scenario. For example, going forward for the zero shot, the one thing I just mentioned above is that there is this challenge about the different ratio of cell types in the huge atlas. So we want to do some hierarchical cell type labeling to make sure that it's performing well in a tree-based you know, reference mapping way, so it can perform well even for less observed cell types. And we also going to provide some new checkpoints specifically for zero-shot cell embedding. And we're also trying to explore the space of, you know, more space about zero-shot, but for gene-related applications using the zero-shot gene embeddings, and also other things as well. Yeah, I think this concludes my my talk. Let me go back to this one. Maybe you're interested in demo. Yeah, just one thing. You can also download the output cells and things like that. Yeah, let me see if there are any questions related to any part. Yeah. So Burke, do we have to stop right at?
No. If anyone has any questions, feel free to just unmute yourself, and if you're feeling shy, go through it through the chat. But we can have another 5 minutes, so I'm on a rush.
Thank you. Yeah. So I will first try to answer some questions in the chat, but feel free also unmute yourself. I think the question right now is about potentially is the input data raw count matrix. That's a great question. We do work on raw counting matrix, you know, previously, and then we found there's a technique we introduced ourselves called value binning. It's like you rank the raw counts in each cell and try to project them into a constant relative values, like from 0 to 50. You can imagine you do so. It's like a histogram, right? And then you can have the bins in the histogram, and then you say, okay, The first one is 0, the second one is 1, and then projecting them to 0 to 50. The thing is that you can find in pre-training, in fine-tune, this is not something that are required for the model. You can pre-train on raw count. You can also pre-train on log normed counts. It's just that we found that the spinning we introduced is actually very beneficial so far for both pre-training and a lot of fine-tuning tasks. You can find more details in our manuscript. What is the running time required to pre-train scGPT on a data set of 33 million cells? When we train it, we took, I think, around 8 days on 16 A100 GPUs. But that's not for one epoch, that's for 6 epochs. So you can roughly finish one epoch of training of 33 million cells in 1 day or so on 16 A100. Honestly speaking, so far, I mean, if you train today, it might be faster because PyTorch actually updated itself. It's more efficient now. How do you deal with the imbalanced data, like far more normal cells than tumor cells, which affects the clustering a lot? I'm imagining you're talking about the zero-shot application. Let me go back to the slide. Yeah, if you're talking about this one, so I just want to clarify first. So the pre-training, we only train on normal cells, but here we apply on normal and disease cells. It looks like in disease samples, and they contain normal and disease cells, right? It looks like the tumor cells. Actually, the model also captures the difference between tumors and others, right? Yeah, and for this one, if it's not in training, you don't need to worry about the imbalanced issue because the model generates the cell embeddings on a per cell basis. But if you're talking about pre-train and fine-tuning, that would be a great question. Depending on what you want to do. For example, if you wanted to fine-tune the model to do some cell type classification, it makes very much sense to do some balanced sampling of your data, or to append different weightings for the, when people train the cross entropy, you can append different weighting for your for the different classes, right? And you can also do that, yeah. How does what you have shown relate to the perturbation prediction? Is this using the same model and training set, or did you use a different training set? We use the same model, the pre-trained model, but that's in a fine-tuning setting. So we fine-tune the model a little bit on the different training set. Let's call it fine-tuning set. And specifically, we use a lot of the perturb-seq data set. So in the perturb-seq, you have control cells, and you also have the observed response, after the perturbation. Yeah. So that's how we approach the perturbation so far. Would there be something similar to hallucination? This is a great question. I received it a lot, but I would be totally honest with you. I think the hallucination is not very well defined so far in the domain. For example, and also, for example, would you consider some error in classification as hallucination, or would you consider something more tricky? You see my point here, right? If it's like some conventional errors, it's not a question specifically related to large language model. All models can make errors, right? But if it's for large language model, maybe you're talking about sometimes you generate a cell like looks like one, but not exactly. Yeah, we honestly speaking, I don't think we have extensive evaluation or definition for this question, but it can definitely be interesting. I think also just another sentence. I think Andrew Kapasi, also recently have a great post about hallucination, but that's related to the general domain in his Twitter. I think it's also very inspiring for the question in general. Yeah. Can you speak?
Do you wanna wrap up with the last question, or?
Yeah, let me see if
okay. And I'll share your email if you're okay with the large group afterwards, and they can shoot you an email if they have any more questions.
For sure. Yeah.
Okay.
Yeah. Just want to say, yeah, I see a lot of potential new tasks or new application like IPSCs and other stuff. Yeah, I think it's also promising to that. I think we definitely can connect more either in GitHub and or via email. Yeah. Thank you guys. I wish you this is a talk for that we can learn something for all of us. Yeah, and hopefully you can enjoy our model. Thank you.
All right. Thank you so much, Atian. Thank you, everyone. Thank you for being here. Have a great week. I'll follow up with the recording if you want to watch it again, and feel free to reach out to me, to Atian, with any questions you might have. Have a great week. Cheers.
Thank you.